Hello and welcome to this Introduction to Microbiology video. I'm Dr. Aled Roberts, and in this video we're going to look at the key quorum sensing system in Staphylococcus aureus, from how it works to its effects at high cell densities. Now, during infection, Staphylococcus aureus utilizes a single quorum sensing system to help regulate things like virulence factor production, biofilm formation, and the overall colonization of a host. Essentially, this quorum sensing system allows bacterial cells to assess their population density and adjust their behaviour accordingly. Now, the main quorum sensing system of Staphylococcus aureus is the accessory gene regulator, or AGR system. This system enables Staphylococcus aureus to detect and respond to its own population, functioning as a mechanism for intra-species communication. Now, there is a second system known as the autoinducer 2 system, which is based on the Lux quorum sensing system seen in other bacterial species and serves as a universal signaling molecule, allowing Staphylococcus aureus to sense the presence of other bacterial species, functioning as a mechanism for interspecies communication. And so quorum sensing helps Staphylococcus aureus navigate complex microbial environments and potentially coordinate behaviours with or compete against other microorganisms. Now, for the rest of this video, we're going to go on a deep dive into the key quorum sensing system in Staphylococcus aureus, which is our AGR quorum sensing system. Now, the genes required for the AGR system are encoded on the AGR operon, which we can see here. And there are 4 genes with a single promoter region known as P2 that drives the transcription of the RNAIII transcript, which encodes for the AGR A, B, C, and D genes for this quorum sensing system. And so under low cell density conditions, transcription factor AGRA associates with the P2 promoter region of the AGR operon and transcribes the operon, producing the RNAIII transcript. This results in what we call a basal level of transcription that will ultimately result in low levels of each gene. Now, one key product of the AGR operon, which we have just seen in action, is actually AGRA, which is a transcription factor protein that helps to promote the production of many genes, including the AGR operon, as we can see here. Now, another product of the AGR operon is AGRB, a transmembrane channel protein that embeds itself in the cell envelope and has 2 main functions. The first is that it acts as a transporter, allowing components to move between the cytoplasm and the external environment, which might not be able to otherwise. The second function is that it possesses enzymatic activity, meaning it is able to modify and process molecules as they pass through the transporter. Now, the third product of the AGR operon is AGRC, another transmembrane protein but with a slightly different function. You see, this embeds itself in the cell envelope and has 2 functions. First, it acts as a receptor for the AGR quorum sensing system, detecting the presence of its corresponding autoinducer. Second, it functions as a histidine kinase, meaning upon binding to the autoinducer, it undergoes autophosphorylation. This phosphorylation event is a key step in initiating the downstream signaling cascade that regulates gene expression. Now, this brings us to the final component of the AGR operon, which is AGRD, and this serves as the autoinducing peptide, the key signaling molecule for the AGR quorum sensing system. Now, when AGRD is first synthesized, it's translated as a long polypeptide chain of around 45 to 47 amino acids, and it's in an inactive form, and so at this stage it's referred to as a pro-autoinducing peptide, as it requires post-translational modifications to become fully functional. And so to undergo this essential processing, the pro-autoinducing peptide must be transported to the cell surface. When it gets here, it will interact with the transmembrane protein AGRB, and at this stage, AGRB performs 3 critical functions. First, AGRB activates its endopeptidase activity, cleaving the pro-autoinducing peptide to release a short peptide of around 7 to 9 amino acids in length. Second, AGRB facilitates the cyclication of this peptide, and it does so by forming a thiolactone bond between a central cysteine residue and the newly freed carboxyl terminus, creating a stable ring structure. Now, this circularization process is crucial because it confers bioactivity to the molecule, and so at this point it is officially recognized as a fully functional autoinducing peptide capable of participating in quorum sensing. The only thing left for AGRB to do is transport the mature autoinducing peptide out of the cell and into the extracellular environment, where it begins to accumulate as cell densities increase, setting the stage for activation of the AGR system. Now, at low cell densities, the concentration of the autoinducing peptide in the environment remains relatively low. However, as cell density increases, so does the accumulation of our autoinducing peptide molecules in the extracellular space. Therefore, at high concentrations of cells, the autoinducing peptide will start to accumulate, and once this minimum activation threshold is met, the autoinducing peptide molecules will begin to interact with AGRC, which, if we remember, is our membrane-bound histidine kinase enzyme. Now, this interaction triggers AGRC activation, allowing it to become autophosphorylated. This essentially means AGRC utilizes cytoplasmic ATP, breaking it down into ADP and inorganic phosphate, with the ADP being released back into the cytoplasm for recycling, while the phosphate group is retained on a conserved histidine residue within the AGRC protein. And so at this stage, AGRA, which if we remember is our response regulator and transcription factor of the AGR operon, can associate with the phosphorylated and activated AGRC, accepting the phosphate group through an exposed aspartate residue. Now, what this does is it causes a slight conformational change that increases the overall activity of the AGRA protein, allowing it to participate in a number of signal transduction cascades, where it helps to turn certain genes on and off. Now, one of the first things it does is what it has been doing all along, associating upstream of the P2 promoter for the AGR operon. And due to its activated state, it has a much higher binding affinity, leading to increased transcription of genes within the AGR operon above the basal transcription rate we saw at the start. And this essentially means we get more of our transcription factor being produced through the upregulation of AGRA, we get more of our transport proteins being produced through the upregulation of AGRB, we get more of our histidine kinase being produced through the upregulation of AGRC, and finally we also get more of our autoinducing peptide signal being produced through the upregulation of AGRD. And so again, what we see here is essentially a positive feedback loop, resulting in the increased production of our AGR quorum sensing system components. Now, in addition to its increased binding affinity for the P2 promoter, phosphorylated and therefore activated AGRA also increases its binding affinity for the divergently encoded promoter region known as P3. Now, this binding event initiates the transcription of RNAIII, a key regulatory RNA that activates the expression of virulence genes that were previously repressed. And we can think of RNAIII as the primary effector of the AGR quorum sensing system, functioning as a global regulatory molecule that modulates the expression of numerous virulence factors, including toxins, exoenzymes, and surface proteins. And so what this does is it ensures that only specific genes are switched on at high cell densities, allowing Staphylococcus aureus to coordinate its pathogenic response effectively. Now, if we examine this process in a little more detail, when cell density is high, the environmental concentration of autoinducers also increases. This leads to global changes in gene expression, resulting in the upregulation or downregulation of specific genes within the bacterial cell. And so if we look at Staphylococcus aureus under high cell density conditions, we see an increase in the production of several key virulence factors. The first being delta hemolysin and alpha toxin, which are pore-forming toxins that disrupt host cell membranes, cause cell lysis, and help Staphylococcus aureus to evade the host's immune system. We also have an increase in the expression of phenol-soluble modulins, which are multifunctional molecules acting as both pore-forming toxins and pro-inflammatory agents, contributing to host tissue damage and persistent inflammation. Now, at the same time, Staphylococcus aureus downregulates several genes involved in adherence and immune evasion. The first is fibronectin binding proteins A and B, which facilitate attachment to host cells and extracellular matrix components like fibronectin, and so their downregulation signals a transition from a surface adherent lifestyle to dissemination and invasion. Next, we have downregulation of the Staphylococcus protein A, which is a cell wall associated protein that binds immunoglobulin G, also known as IgG. And what this does is it stops opsonization and phagocytosis. And again, reducing SPA levels allows Staphylococcus aureus to shift from immune invasion towards active invasion. And finally, we have the downregulation of biofilm associated genes, supporting the transition from colonization to dissemination, allowing the bacteria to spread within the host. Now, in addition to this, we have other levels of control, and so we also see a downregulation of the ROT gene, which stands for repressor of toxins. And it does what it says on the tin. It represses the production of toxins. However, its downregulation lifts this repression on several virulence factors. And so this results in the increased expression of proteases, lipases, and ureases, enzymes crucial for the degradation of host tissue. And in addition to this, the downregulation of the ROT gene leads to the increased production of various toxins, enterotoxins, and superantigens, all of which contribute to host cell damage, immune system disruption, and systemic infection. And so ultimately, the AGR quorum sensing system in Staphylococcus aureus drives a major regulatory shift, promoting the upregulation of virulence factors essential for tissue invasion, immune evasion, and host damage, while simultaneously downregulating colonization factors. And so this transition enables Staphylococcus aureus to move from an adherent biofilm forming state to an invasive and pathogenic phase. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.